Hello everyone, my name is Gary O'Mealy, and in this video we are going to talk about signal transduction. So you'll recall from one of our previous videos that just about every kind of cell signaling process is going to involve a signaling molecule, which we can use the umbrella term ligand, and that ligand is going to travel either over a short distance, over a long distance, through a variety of different liquid mediums, to a target cell that possesses the correct receptor for that particular ligand. So the idea here is that once the ligand engages with and binds to the receptor, we say that the receptor is in an activated state, and this is in some way going to facilitate changes inside the cell that will eventually produce a physiological response, whatever that may be. Now, the way it's illustrated in this slide, this is going to seem very much like a black box. Just you put the ligand here and then boom, you get your response, right? Well, there's actually quite a few different chemical processes that are going to occur from the time the ligand binds to the time you actually get a response. And oftentimes those chemical reactions are going to depend on what the ligand is and what the receptor is. So our goal here through this video and the next few videos is to orient ourselves to what those types of responses are. So some possible end responses before we actually get into the signal transduction part of this in terms of how does ligand binding actually produce a response. Some possible responses that you could get are, number one, a new protein is made. So the idea here is that whatever this new protein is, it was not being made before, so the new function of the cell, the physiological response, is due in part or in total to whatever this new protein is doing. So the idea here is that activation of the receptor in some way leads to the transcription of a gene or multiple genes into messenger RNA, and those messenger RNA molecules are put through the translation process. New proteins are made, those proteins start doing their function, and because those proteins were not being made before, that's your physiological response. The fact that this cell is now producing proteins that it was not making before. Another possible response is that the target cell is induced to enter into the cell cycle. So you start getting cell growth or differentiation or something like that. You often see this with stem cells. So here, the activation of the receptor is either going to produce some combination of activating proto-oncogenes that get the cell into the cell cycle and or inhibiting tumor suppressor genes that prevent the cell from going through the cell cycle. So this is all about kind of providing the stimulation that is necessary to tell the cell that conditions are favorable for entering the cell cycle, so let's go ahead and do it. Another possibility that is very common is that you will see an enzyme inside the cell is activated because it was in a previously inactive state, or an enzyme that was in a previously active state is induced to become inactive. So it's kind of an either-or proposition. So we've learned before about what enzymes do, and all the metabolic reactions that happen inside of cells in the body are catalyzed by enzymes, obviously, and basically we can see here that through cell signaling processes, the body has a great deal of control over which enzymes are doing what and when. Another possible response is that you can induce secretion. So in this case, activation of the receptor induces the target to begin secreting either a protein or a lipid product that is sometimes synthesized ahead of time and stored intracellularly in vesicles, and you'll often see this in both exocrine and endocrine processes. So exocrine, we're referring to things like sweating. Obviously, you can be induced to sweat as a response to your body temperature getting too high. Exocrine functions are obviously important for the digestive system, and in the very near future, we're going to start talking a lot about endocrine functions and how the secretion of one hormone binding to a receptor will actually stimulate another hormone to be produced and secreted next in line. You can also get the opening and closing of an ion channel, which we will see plenty of examples of very soon, and this is going to actually be very important for the function of the nervous system and for the contraction of muscles, which we will look at in the fairly distant future. So here, activation of the receptor leads to the opening of an ion channel that was previously closed. Vice versa is also possible too. You could have a channel that is open for business, and activation of the receptor causes that channel to close. And finally, you can have the mobilization of an internalized carrier. Now, this is a much more niche kind of process. You're not going to see this quite as often. But in this case, some carriers for polar solutes like glucose or water are sometimes kept hidden from the membrane until the appropriate signal is received. We're going to, in this course, become acquainted with this kind of response when we talk about the actions of the hormones insulin and antidiuretic hormones. So you can look forward to those discussions in the future. Okay, so now that we've looked at the possible responses that we can get as a result of ligands binding to receptors, let's talk about what happens in between there. So that is all about the signal transduction process. This is the method by which a cell produces a desired response from that particular external signal, whether it's a hormone, neurotransmitter, or whatever. So these methods of signal transduction are necessary for any process that utilizes a membrane-bound receptor. And obviously, in previous lessons, we've talked about the difference between membrane-bound receptors and internal receptors. Signal transduction is not quite as necessary for internal receptors because the signal, the ligand, in those cases is hydrophobic and can cross through the membrane. So signal transduction is mostly going to be necessary for ligands that cannot pass through the membrane. So think of it this way. The signal, the ligand, cannot cross through the membrane, but at the same time, that signal is carrying the message that we want to convey across the membrane to all of the proteins and components inside the cell. So the message stays outside the cell, but the machinery for producing the response is all inside the cell. So we have to have a methodology for imparting the contents of that message across the membrane. We have to make sure all the machinery inside the cell gets the message, and that is what signal transduction is all about. So, like I said, if the message can't cross the cell membrane, there has to be a way of conveying the contents of that message to everything that is inside. Okay, let's take a look at what this might look like. So we know for sure that engagement between the ligand and receptor is necessary to produce a response. So in this illustration, we have an extracellular ligand that is not going to cross the membrane because it is hydrophilic, but it does have a membrane-bound receptor here, and we can see how the ligand is going to bind. But we can see eventually that the response is going to actually happen in the intracellular side, not the extracellular side. So ligand is going to stay out here. Somehow binding of the ligand to this receptor is going to produce a response inside here. So let's take a look at how that happens. So the receptor does not exist all by itself. The receptor is often complexed with another component called a transducer and another one called an amplifier. For the sake of simplicity here, I am showing the transducer and the amplifier as being one and the same, but that is not always going to be the case. So the idea here is that, in particular, the transduction and amplification portion of this is going to lead to the production of what are called second messengers. Second messengers are chemicals that are rapidly produced inside the cell not long after the ligand binds to the receptor. Very often, these second messengers are produced by enzymes that are activated through the transduction process. So the second messenger is essentially going to carry the same message that the original ligand is carrying, just with additional weight. Now, what makes me say with additional weight? Do you notice how there's only one ligand, but there's multiple second messenger molecules? So essentially what's happening here is that we have not only conveyed the contents of the message inside the cell, that's the transduction portion of this, but we have amplified the signal too. So we can produce a very strong and significant response inside the cell with a very small amount of ligand. So we can produce a lot of second messenger with a very small amount of essentially what we call the first messenger, the ligand. Now, what this second messenger is often going to do is activate target proteins inside the cell. A lot of target proteins have binding pockets for second messengers, and we'll learn about what these second messengers are in future videos. But obviously, the binding of these second messengers, you can see, is going to change the function of these target proteins, and that in some way is going to produce a response. Now, this is still somewhat vague in terms of exactly what's going on, but this is a very good first step in starting to understand how signal transduction works. And when we start talking about very particular and very specific signal transduction cascades, we'll get a better idea of exactly what's going on here. So this is just very general for now. So I mentioned that there are several different types of signal transduction pathways that we're going to be looking at in this course. So the type of pathway we're going to utilize is very much going to depend on what kind of receptor we have and what kind of ligand is binding to. But in general, we can split it up into three different categories. We can have the activation of what is called an ionotropic receptor. This is a very, very simple process in which the receptor itself is actually an ion channel that will open up or close depending on whether the ligand is present or not. We'll go ahead and take a look at this now, but for now I do want to mention the other two that I mentioned, which we will cover in future videos, which are G-protein coupled receptors and tyrosine kinase receptors. So we will talk about those two in future videos, but for now, let's go ahead and finish this video off by showing you exactly what we're talking about with ionotropic receptors. So look at this situation here. This is a sodium channel right here. You'll notice that this sodium channel is currently closed. Now, we, of course, know from previous lessons that there exists a very strong concentration gradient for sodium across the membrane, in which sodium is maintained at a relatively high concentration in the extracellular fluid and a relatively low concentration in the intracellular fluid. So the sodium in the extracellular fluid wants to move downhill across the membrane to go inside the cell. But the thing that's currently preventing that right now is, of course, the very fact that sodium is electrically charged and cannot move through the membrane all by itself. If sodium is going to move, it needs an open channel or carrier, but as it currently stands, this channel is closed. So sodium, even though it wants to move across the membrane, it can't do it right now. But let's see how things change when ligand becomes available. You'll notice that this ion channel here has binding pockets for a ligand. So if this ligand were to bind into those binding pockets, the channel is induced to open so that now sodium can move down its concentration gradient from extracellular fluid to intracellular fluid. The consequences of the opening of this kind of channel is something that we will explore when we talk about the nervous system, but you can go ahead and take it to the bank right now that the best example of this type of response is what happens when acetylcholine, a neurotransmitter, binds to nicotinic receptors. Nicotinic receptors are what we call ionotropic receptors because the receptor for the acetylcholine is an ion channel itself. So when acetylcholine does its job, you'll know that the response is that you open an ion channel. Okay, we'll go ahead and stop there for this video. Here's a list of vocabulary terms you probably ought to be aware of. And for checking your understanding, number 1, why is signal transduction necessary for hydrophilic ligands? And number 2, which component of a signal transduction pathway allows a very strong response to be produced by a very small amount of external ligand? All right, that's going to do it for this video. If you have any questions, go ahead and drop them in the comments section, and I will see you next time. So long.